Good afternoon, everyone. I'm Fana Salehi, first-year Ph.D. student in Dr. Gerton lab. I'm honored to introduce Dr. Gerton to you all. Dr. Gerton is an investigator at Stowers Institute for Medical Research, investigator of cancer biology, University of Kansas Comprehensive Cancer Center, and professor at Department of Biochemistry and Molecular Biology, University of Kansas Medical School. She obtained her bachelor degree in human biology and a Ph.D. in microbiology and immunology from Stanford University. She became a part of Stowers Institute in 2002. She focuses on studying chromosome biology, genome and maintenance and stability, and the formation of Robertsonian chromosomes. For much of her career, she worked on questions about the basic biology of chromosomes using yeast as a model, but in recent years has transitioned to study genome stability in many organisms, including humans. Dr. Gerton has received several scientific awards, including the University of Kansas Cancer Center Directors 2022, Basic Science Award, Center for Excellence in Research, Cornelia de Lange Syndrome Foundation from 2014 to present, and the Hudson Prize in 2007. Let's give a warm welcome to Dr. Gerton, who is here to shed light on illuminating the dark matter of human genome with us.
Thank you, Farnaz, for that introduction, and it's a pleasure to be here today. Thanks for the invitation. It's been great to meet a few of the faculty and the students today, and I look forward to more interactions throughout the day. So, if I forget to advance my slide, please tell me, because I've got two systems going up here. Okay. So what my lab is interested in is the genetic and epigenetic variation in genomes and how that impacts the biology of chromosomes. And so today I'm going to focus on the human genome, but as Farnaz mentioned, we have other projects in the lab where we study yeast and zebrafish and mice as well. So I thought I'd just take a second. Let's see. Ooh, why is this not going? Maybe here. I tried that, didn't work. The clicker. Let's try the clicker. There we go. Okay, great. So I thought I'd take a second and tell you a little bit about who I am and why I do what I do. It sounds like there's a number of people here interested in genome integrity, but we know that genome integrity is really important, and without it, there are lots of pathological consequences. So I'm a chromosome biologist, and I've been studying genome integrity in many different forms for most of my career. If you compromise genome integrity, what can happen is it can have human health impacts. So it can cause things like, or it can underlie diseases like cancer, infertility, some specific rare diseases. And so this has motivated my interaction with a number of different organizations throughout my career, and a lot of those are listed here. And in particular, my interaction with the Cancer Center and various cancer research organizations. But I think really important for the talk I'm going to tell you about today, I've been interacting with this telomere to telomere consortium, or T to T, as I'm going to reference it, that's been integral in assembling human genomes and other genomes from telomere to telomere in a complete form for the first time in human history. And so that's really motivated a lot of the work that I'm going to tell you about today. But I think it's important that we understand the basics of genome integrity and how the integrity of the genome is maintained, because I think with that type of information, someday we can harness it to have impacts on human health. Okay. So this is a quote I like. I don't know if Einstein really said this, but he said that the work of the individual is really bound up with that of his or her scientific predecessors and contemporaries, and it almost appears as an impersonal product of your generation. And so we all in this room, we work in the current, scientific milieu. We work with our current technologies. We work in our current paradigms, but we're building on the shoulders of all the people that came before us. And there were a lot of significant events that happened, in genome science during my lifetime. And I talked a little bit about this with the students. So, when I was at Stanford, I was a PhD student. The very first eukaryotic genome was being assembled at Stanford. I wasn't part of that, but it got me really excited about budding yeast as a model system. And I was in the lab, one of the labs where DNA microarrays were being invented. And so that really got me thinking about the whole genome, not just, gene structure function types of studies that had been more of the norm until then. And I think that, back in 2003, the very first human genome, reference genome, was produced. And that actually was sort of the starting point for a real explosion in development in sequencing technologies that led us to where we are now, where we can really start to really completely assemble these human genomes. And the two stories that I'm going to tell you about from my lab today are kind of built on top of this foundation, this baseline that is being established by this really worldwide group of scientists. Okay. So my seminar is going to have two parts. The first part, I'm going to talk about centromere variation and how that impacts centromere structure and centromere function. And then the second part, I'm going to tell you about a type of structurally variant chromosome called the Robertsonian chromosome, and some new work that we think starts to explain how these form in the human genome. Okay. So what are centromeres? It sounds like Glenys Logston visited, so you've probably had a primer on this a little bit. But centromeres, when you look at chromosomes, they're often referred to as the constriction. So it's the part that kind of like the narrow, skinny waist of the chromosome. And these are genomic loci that are present on all chromosomes, and they're really important for the transmission of the chromosome from cell to cell when the cell divides. In this particular diagram, the centromeric DNA is represented in this green color. And centromeres have two, I think of them as having two really important functions. One is it's the location where the kinetochore gets built, and that is signified by this blue ball here. And that is where the microtubule is attached to the chromosome to pull the sister chromatids apart when the cell divides. And then the other important feature of a centromere is that it actually holds the two sister chromatids together until the chromatids are bi-oriented, and in order to ensure the accurate segregation. So that's represented by this pink color here, and that's in large part mediated by a specific protein complex called the cohesin complex, and it really is genuinely thought to form a ring structure that literally embraces two pieces of DNA, and that's how it holds the sister chromatids together. And I would say decades of research, some of which from my lab in budding yeast, has gone into understanding these protein components of the centromere. So understanding the microtubules and the kinetochore and the cohesin complex. But I think what has been much less well studied is the role that the centromeric DNA itself may play in this process. And in part, that's because we haven't had access to that information. That's part of the dark genome. So these linear sequences of centromeres, we haven't known what they look like. And this here is just a video to remind you that chromosome segregation is a very dynamic process. The centromeres are labeled here in green, and you can see that they kind of like jiggle around to get all aligned and bi-oriented. And there's actually some stretching motions if you follow one carefully. And then the sister chromatids come apart very beautifully. And so we wanted to understand, what's going on at the centromeric DNA during this process, and what's going on with stretching. And we wanted to understand what's going on with sister chromatid cohesion. And could we understand this in a new way based on our newfound access to the centromeric DNA sequences themselves? And so I'm going to tell you a little bit about that centromeric DNA that we kind of just are starting to appreciate what it looks like in humans. It's highly repetitive alpha satellite DNA. It's composed of these 171 base pair repeats that are in this higher order array. And it can be very variable in size. So it's very polymorphic between chromosomes, between individuals, but even between haplotypes. It's actually quite refractory to recombination. So it's inherited in these gigantic megabase size haplotypic blocks. I'm going to tell you a little bit more about that. And there's some portion, some sub-region within this giant repetitive array where SEMP-A binds. And that is a specific histone variant that specifies the centromere. And that's where the kinetochore gets built. So this is like the kinetochore forming function of the centromere right here. And I want to emphasize how polymorphic in size centromeres are, because it's important for this talk, that these arrays, even between like chromosome 1, could vary in size by as much as 5-fold. So it could be 1 megabase or it could be 5 megabases of DNA. And when you think about the function of the centromere, you imagine that could actually impact its function. All right. And as I mentioned, each of us has our own unique collection of centromeres. So you inherit, it's a haplotype, you inherit centromeres on the chromosomes from your mom, and you inherit a collection of centromeres from your father. And then together, those make your very unique collection of centromeres in your genome. And we got very intrigued by the possibility that each person's unique collection of centromeres may impact their predisposition to, let's say, aneuploidy or genome instability, or and the knock-on effects that this may have for human disease. So that's a very big question, and I'm not going to answer that question today. But what we have done is kind of broken it down into a couple of smaller questions that I am going to talk about. That is, how do these big variations impact the structure of centromeres? And then furthermore, how do they affect the performance of those centromeres when it comes to actually segregating the chromosomes? And I want to mention very much the people who are responsible for this work. So this work has been led in large part by this terrific postdoc in the lab, Ayantika Sengupta. And we work at Stowers. We're very fortunate that we have a lot of PhD level scientists in our core centers. And these are some of the people who were really instrumental to the work that I'm going to show you today. We have people in bioinformatics and we have people in microscopy. And without them, we could not have done this project and our funding from the NCI. And part of this work was recently published and part of it is still unpublished. And I'll get to that in the second part. So to begin to look at the structure of human centromeres, we wanted to just do a very basic kind of experiment. That is, where are these proteins located relative to that big piece of centromeric DNA? Where's SEMP-A? Where's cohesin? And we did that by ChIP-seq in the beginning. And then we moved on to use super resolution microscopy to look in a more 3D visualization. So the first thing we did, though, was this ChIP-seq approach. And I'm going to show you that data first. So we actually were very fortunate. Glenys Logston gave us access to her SEMP-A ChIP-seq data. Remember, that's the histone variant that specifies the centromere. And I'm showing you now chromosome 18. And the part here in green is where the alpha satellite DNA array is. So you can see that SEMP-A maps really nicely to this alpha satellite DNA array on centromere 18. And you can even see this subregion of much higher enrichment, like I showed you earlier in the cartoon. So using those exact same mapping parameters, we wanted to see, where does cohesin bind? And we were very fortunate that we could do this experiment in the same cell line that we have the reference genome for. And so these are 3 different subunits of the cohesin complex. And I think what really jumped out at us right away was it does not map in the alpha satellite DNA. So cohesin does not map to the alpha satellite DNA. Instead, it's in these regions that are pericentromeric. But it does map within, so I didn't tell you this gold bar up here is actually a haplotype. That's a centromere haplotype. So the haplotype contains the information for the binding of both of these protein components that are essential for centromere function, the cohesion function, and the kinetochore building function, but they are physically separated in linear space, at least, from this. So that was something we didn't know before. I'm showing you centromere 18. This was the same across all of the centromeres. We did this in 2 different cell lines. This is very much the pattern that we observed. So the next thing we wanted to do is try to understand this in 3 dimensions. So we wanted to look at these same protein components relative to the centromeric DNA, and we use super resolution microscopy. So what I'm showing you here is the mitotic chromosome spread, where you can see the location of SEMP-A and cyan. There's an antibody staining, and then the location of RAD21 also by antibody staining in magenta. And this is just kind of your standard microscopy, but then Iontica wanted to use super resolution microscopy to really get in and look at that centromere region in particular. And I think what you can appreciate from these are kind of zoomed in versions on the centromeres. It's a bit pixelated, but you can see that the SEMP-A domains, which is represented in cyan, and the cohesin domain in magenta, they are not, they're not together. They're clearly non-overlapping in 3D space. So they're non-overlapping in 2D space, they're non-overlapping in 3D space. This was consistent with our ChIP-seq, but it also kind of advanced our understanding of how these things might be arranged further in 3D. And so the next thing she did was she collected 400 images of chromosomes, and she did something we call SPA-SIM, and that's single particle averaging of super resolution images. And she aligned and summed up all of these images with the help of our great collaborators at Stowers, and she was able to find essentially what is this common core structure amongst all of the centromeres in these 400 images that she collected. And this is a lot. I have a lot of technical details about how she did this, and I'm not going to go through these today, but I just want to, so this was a very technically rigorous study. And what I do want to show you is that there was this common core geometry where SEMP-A, the 2 SEMP-A domains were the same distance apart or very similar. And then there was this core of cohesin that is parallel to the axis of the chromosome. So if the chromosome, you could see the chromosome would be kind of stretched this way. So we thought that was interesting because we know that the DNA can be very different sizes, but we're, at least with the protein components, we're achieving this common core geometry. We thought that made some sense because all the centromeres have to be recognized by the mitotic machinery. And so maybe they like to have, you know, a common core structure that they're recognizing. But it kind of begs the question, like, how are the really, like, the 5 megabase centromeres versus the 1 megabase centromeres, how are they being organized? So the next thing we wanted to do was take this information we had, and we started to build a working model for how the DNA might be organized at the centromeres. And here we've got 2 sister chromatids. They're being held together by our ring cohesin complex. So that would be this core, this axis, this axial core. And then part of this model is we think that alpha satellite DNA is being extruded as a loop. And if you work in the cohesin field, loop extrusion is a big topic right now. So we think this is the alpha satellite DNA. We think it's being extruded as a loop, and then the SEMP-A domains are out here at the edges. And this would be intramolecular, and this is intermolecular. So this is just a working model now that we can use to kind of build on to try to understand how the centromeric DNA is organized. But we still really wanted to get a sense of how these very different sizes of DNA could be accommodated in this structure. And so it turns out that we can detect these different haplotypes in our super resolution microscopy images, and that turned out to be a really useful tool for us. So you can see here, this is a FISH, fluorescence in situ hybridization probe. This is a FISH probe to SEN7. And one of the haplotypes, one of the centromere haplotypes is very bright, and one is somewhat dim, and we've quantified that in many, many cells. So we can actually distinguish the bright and the dim haplotype in our super resolution microscopy. And then we can ask, how is the DNA organized differently? And I'm showing you this for Sen7. We're now doing this for many more centromeres. And then what she does is within a single cell, we've got our dim and our bright haplotype, and then she divides them up into the dim and the bright for each cell, and she performs that same SPA-SIM, so single particle averaging of these images, and we can come up with like an average image of the dim and the bright haplotype. And that's what I'm showing you here. And you can see what the DNA is doing now. So here in the dim haplotype, the DNA is really just stretched right across the center. In the bright haplotype, it's kind of spreading a bit more along the chromosome axis. And now I've got my chromosome over here to orient you. But it turns out the SEMP-A distances are still very similar. And so it seems like what's happening is that alpha satellite DNA is being packaged somewhat differently in order to maintain this common core geometry. Okay, so what our current working model is, our current hypothesis is that these alpha satellite DNAs here the loops can be bigger or smaller and spread along the axis in order to accommodate whatever alpha satellite DNA happens to be there. And we're very curious to know kind of how these looping events occur. We don't know that yet. The next thing she wanted to ask is whether these two centromeres have any functional differences. They look a little bit different. And so the paradigm that we've been using in the lab is a paradigm that is called cohesion fatigue. So essentially what you do is you take cells and you hold them in metaphase for a long time with microtubules pulling on the centromeres. This was something that was first and if you hold those centromeres under tension for long enough, eventually they'll come apart. And we wanted to know if the dim haplotype came apart sooner or later than the bright haplotype. So that's the experiment that she did. And she's measuring, whether the two centromeres are together or apart. And you can see here actually that the dim centromere comes apart earlier than the bright centromere, than the bright haplotype. And so we're doing this now for multiple centromeres. That's shown here. We've actually looked at even more now, and this is and two different cell lines. So this is definitely the trend that we see. The dim haplotypes are coming apart sooner, and we think that's interesting because if sister chromatids come apart too early, what can happen is those sister chromatids can missegregate. So the next thing we need to do is actually look at chromosome missegregation as a function of haplotype differences, and that's in the works right now. Okay. So then just to sum up this first part of the talk, how do unique centromere haplotypes, how do they impact centromeres? I think one thing we can see is that they impact centromere structure to some extent. There is this common core. The alpha satellite DNA is where it has the kinetochore function. The sister chromatid cohesion is in these pericentromeric regions. I forgot to say, but this is actually quite satisfying in a way in terms of the model because it allows that alpha satellite DNA to stretch and sister chromatid cohesion to remain intact. So that's actually been quite a puzzle in the field. How is that DNA stretching, but the sister chromatids stay together? It's because it's two different domains of the centromere. And then depending on the size of your array, it can spread, it can be packaged somehow differently. And we're very curious to know what some of those organizer proteins are. And then in terms of the performance of these different centromere haplotypes, we've seen these differences in fatigue under kind of stressful conditions. It could also be like, we're going to look under things like if we have P53 mutation, some kind of checkpoint error, correction is not intact. And will that lead to aneuploidy? And will, let's say, a dim or a bright haplotype be more prone to generating aneuploidy? So that's all in the works. And I think what I'm going to do now is I'm going to take a short pause and see if there are any questions on that first part of the talk. Yeah.
So have you just looked at cancer cells to see if they're surveyed and see if there's difference in haplotype in terms of if you compare it to the two in terms of haplotype, you determine whether there are overrepresentation or underrepresentation of bigger haplotypes in comparison to the final two in cell.
I think that is kind of where we're
Can
headed.
you
We haven't done
repeat the
anything
question?
quite like that, but we would need to know the original haplotypes of the cell and then kind of what's getting missegregated. But I think that's kind of exactly like the long-term vision of where this research could go. Yeah.
Can you hear me?
Oh,
Just
yeah.
remember to repeat the question because some
Oh,
of
sorry. Yes,
on Zoom
sorry.
can't
You
hear
can't
it.
hear the question very
Yep.
well.
Yep.
So the question was, can we see this kind of biased missegregation in cancer cells? And we haven't done that experiment yet. I should say the one cell line that we have looked in, it's a cell line. It's not like a primary tumor. This HCT116, this is a cancer cell line. So the haplotypes are all different. Every time we start this experiment, we have to determine which haplotypes we can distinguish by FISH. It's not the same in every cell. It's actually quite different. But when we can distinguish the haplotypes, then we can do this, ask about this bias.
I guess a follow-up question. So if there is a lower tendency to segregate or it's a delayed segregation, would that favor crossing over events where you're more likely to have a chromosome crossover, or would that be not?
You're thinking now meiosis, I think, because a lot of crossovers happen in meiosis.
I think
Uh-huh.
I guess that's between chromosomes. Whether it is that.
I haven't thought all the way through that, but if you like translocation, stay tuned to the second. Oh yeah, I'm sorry. The question was whether these biases might lead to crossover events. And the answer is, I don't know, but we're going to talk about crossovers in the next part. Yeah. And we'll talk about centromeres in that as well. Yeah.
Some kind of working to keep the two strands together. So when you separate them, both the rope break or the chromosomes break?
Right. So he wants to know where when the cohesin ring has to come apart, how does that work? And there's actually a whole orchestrated group of steps and proteins that have been characterized. So one of the subunits actually gets physically cut, and then the ring opens, and then the one that gets cut gets degraded, and the ones that open kind of stay together. And then in the next round of DNA replication, they can kind of come back on. So people have worked that out. Really beautiful work in budding yeast, and since been validated in mammalian cells as well. But there's a whole slew of proteins I didn't tell you about that do that job. Yeah. Yeah.
Maybe I missed it. What is the role of functional?
What's the role of functional alpha satellite? Well, so one of the interesting things about human centromeres is they don't have to form on alpha satellite DNA. I think nobody really knows why alpha satellite. There's something called a neocentromere, and it can arise someplace else on the chromosome, and there's never alpha satellite DNA where it arises. And I think nobody has really much of any idea why a different region could serve as the centromere. So alpha satellite DNA is definitely not essential for centromere activity. But, you know, some people think maybe 171 base pairs, that's like kind of suggestive of like nucleosomes, like wrapping, but I think we really don't know. Yeah, you've been waiting, so.
I had two questions. One, I'll ask you. Given the connection between centromere formation and chromatin state, especially if you have such a heterochromatic environment, you're getting chunks of centromeric sequences that can fluctuate depending on the parent, the chromosome and the parent. We talked a little bit about this in the cancer genomes, that you argue that you titrate out or release heterochromatin factors. Has anybody looked to see in chromosomes from one parent where you have a larger centromeric sequence versus the other parent where you have a smaller sequence? Is there a correlation with changes to telomeric structure, since a lot of heterochromatic factors are shared between both of those environments? Is there a correlation at all, or have people actually looked?
So the question is whether there's any kind of titration effect with different heterochromatic regions like centromeres and telomeres. And my sense is probably nobody's looked at that at this point. Did you have a different answer? Or? It's interesting. Yeah, I think that.
Those dependent on the genome, then where they go.
Right, yeah.
I guess I'll ask the second question so you can go on very quickly. Suvar 20, I think, K4, K20 transferases have been shown early on by Ray Glasgow. You get decompaction of the centromeric structure. Have you guys looked at all as to whether if you look at specific heterochromatic regulators, are you changing this packaging mechanism but still keeping the spatial structure?
Yeah, so he's asking about heterochromatic proteins and the maintenance of the structure, and we have not done that, but we're very interested in organizer proteins. So we're looking at condensins, I think is one kind of obvious. That's a new PhD student in the lab. We're looking at CEMP-B, which actually localizes across the entire alpha satellite array. And we always get this question about heterochromatic proteins and their localization relative to these particular proteins that I've showed you, CEMP-A and cohesin, and we just don't know the answer. I would imagine like Karen Miga and Glennis Logston are probably going to be doing those kinds of experiments. Yeah. Yeah.
So the cohesin, so it has this impression that it kind of is walking and extruding. So walking along the chromosome pairs quite far. How far out can you see it?
Oh, it's the entire chromosome. It's over the entire chromosome. Yeah. This is like you're asking questions that people have been trying to answer for 20 years, I think. So he's asking where does cohesin start and where does it end and how far does it move? And I think these are all wonderful questions, and there are people working on them. And it's certainly true that in terms of mitosis and meiosis, like the cohesin in the centromeric region is preserved the longest. So all the stuff on the chromosome arms gets, there's like a pathway to get rid of it. And then the stuff at the centromere is maintained. So you may have even noticed that in the images. So if you look earlier, there's cohesin everywhere. But if you wait till mitosis, you really just see most of the signal right at the centromere. And there's maintenance pathways, there's proteins that kind of protect it at the centromere. In terms of loop extrusion, how that works, that's like a whole field. You can go to like a whole conference on how loop extrusion works if you wanted to. So I don't think I could answer that question very well. Okay, this will be the last one.
Like we study ancestry using the mitochondria DNA because it does not go through crossover. So can we understand the ancestry looking at these cohesion because they are not going through the crossover?
Can we understand? I think you can understand ancestry from looking at the centromeric DNA, but I'm not sure how you would do it in terms of cohesin. So cohesin is like, it's not a very specific binder. It kind of binds everywhere. It doesn't bind to any sequence or motif. Well, that's not true. It associates with CTCF, which is a sequence specific binder, but cohesin itself is pretty non-specific. And I think the idea is actually, it's quite dynamic. So it's probably kind of turning over. There's a large amount of it that's turning over very rapidly. And these loops are like forming and then kind of coming apart, and there's beautiful videos on the internet of this. Yeah.
We can study ancestry looking at the centromere DNA.
I think so, yes.
Thank you so
Yeah,
much.
because
Thank
these
you so
are
much.
inherited. Yeah. Okay. So I'm going to move on to the second part of the talk, where I'm going to talk about these structurally variant chromosomes called Robertsonians. And we have some new evidence regarding how they form in the human genome. And I want to make sure that I give a lot of credit because this was a really wonderful collaboration with Eric and Andrea here, as well as Serge and Adam at NHGRI. And then the main players from my lab are Tamara Potapova and Leonardo Gomez de Lima. And it's really been a pleasure to work on this project with our collaborators. So I'm going to start with a little history of Robertsonian chromosomes. They were first described by a guy named Robertson, and he observed them in grasshoppers over 100 years ago. And they've since been observed in many, many species. So you can find them in ruminants. They're actually kind of a problem in terms of the agriculture industry. They've been observed in bats and fish and butterflies and many, many species where people have looked, they've often found these chromosomes. So what is a Robertsonian chromosome? Essentially, it is a chromosome where the centromere is located near the end, and you have two of these chromosomes here, and they fuse together. That's the Robertsonian translocation or the Robertsonian event. These events actually are very important in terms of speciation, and people often refer to this as chromosome speciation. So it's a major form of karyotypic evolution, especially in mammals, and it often leads to reproductive isolation and speciation. So interestingly, the house mouse, Mus musculus domesticus, is really, really good. It's a rock star when it comes to Robertsonian translocations. They're found over 100 distinct karyomorphs have been documented. I'm going to show you an example of that here on this slide. So if you collect, a house mouse from your lab here in Memphis, it would probably have 20. No one has mice in their house, right? Okay, so it would probably have 20 pairs of chromosomes, but if you happen to visit the island of Madeira, there are actually, I think, 6 distinct karyomorphs that have been documented on this one tiny island, and I'm just showing you one of them here. So essentially, these chromosomes can fuse to each other in a translocation. And this is 11 pairs of chromosomes instead of 20 pairs of chromosomes. You can see now the centromere is in the middle. That's called a metacentric, versus these are actually, mouse chromosomes are called telocentric. The human version are acrocentric, and we'll get to that in a second. These chromosomes are also interesting because they exhibit meiotic drive. That means they can be inherited at non-Mendelian ratios. So they can actually be found in higher than Mendelian ratios in the progeny. Sorry, so my daughter texted me, and I'm going to turn that off. So we were very curious why these chromosomes form so frequently, and we decided to tackle this question in humans. And my laptops, I got to plug in. Hold on. One thing we didn't think about when we were rigging up all of this technology. Yeah. Power. This should be fine. So we thought we could figure this out with our friends, with Adam and Eric and colleagues. And we thought if we did that, maybe we would start to understand something more broadly about how these chromosomes form in nature. So in the human genome, these translocations typically form between the acrocentric chromosomes, and those are shown in these green boxes. These are the chromosomes that I love the most: 13, 14, 15, 21, and 22. And actually, it turns out that they don't all form equally between all of the chromosomes. They actually 85% of them, they're called the common Robertsonians, and they involve chromosome 14. They tend to form in female meiosis, not always, but most of the time. And they're pretty common in the human population, about 1 in 800 individuals. So these are all very curious and we don't really know why. In this particular karyotype, what you can see is one of the chromosome 21s fused to chromosome 14. And even though this individual has a normal number of chromosomes, sorry, I'm telling you about my next slide. So in this example, there's a fusion of 14 to 21, but there's still 2 copies of 21. So this individual actually has Down syndrome. So these Robertsonian translocations can have human health impacts, and one of them is their contribution to Down syndrome. They can also contribute to other trisomies like trisomy 13. They can contribute to infertility, and there is a relatively recent paper arguing that carriers have increased incidence of cancer, but nobody really understands why that would be. It's not understood. So like I said, we wanted to work with our collaborators to try to understand this. And I think there were 2, we've worked with Adam and Eric and colleagues on a lot of these projects, but I wanted to highlight 2 that were really important for today. The first being the complete assembly of the CHM13 genome, including the short arms of the 5 acrocentric chromosomes. Those had never been assembled before 2022. That was pretty important foundation for this work. And then Adam and Eric did this really amazing population-based analysis of what was in the short arms of those chromosomes and found something really astonishing and surprising. And what they found were these regions that they called PHRs, for pseudo-homolog regions. So these were regions on the short arms of these 5 acrocentric chromosomes in the human genome that have near identity to each other in the population. And I've shown them as contiguous colored blocks. They actually aren't necessarily all contiguous, but they are quite large regions. So megabase size regions of near identity. And when they shared this with me, I got very excited because I had been reading some old papers on Robertsonian chromosomes, and these common Robertsonians were known to form somewhere between the centromere and the ribosomal DNA repeats, which are also on these arms of the chromosomes. And so, this immediately suggested, by analogy to the PAR regions, that there might be some ongoing recombination between these chromosomes that could potentially explain how Robertsonians form. And so we started to really dig into that idea and how that might work. And what Andrea found, and was also working with the postdoc in my lab, with Leo, what they found was that the chromosomes that commonly form these Robertsonians actually shared a particular subregion of homology, and it was only between these 3 chromosomes. And within that region, there was an enrichment for these PRDM9 sites, which is a meiotic protein that essentially creates an environment that is permissive for meiotic recombination. So that started to really smell like we were onto something about how these recombination events might form. And this enrichment for PRDM9 is found within a particular macrosatellite DNA called the SST1. And these are repeats of the same sequence over and over and over. So this could really be a hotspot for meiotic recombination. That became our working idea, our working model. And interestingly, on chromosome 14, this array was inverted. So if you lined it up with either 13 or 21 and you draw a crossover event, then you could fuse the long arms of the chromosomes, and that essentially would be what has been observed as a Robertsonian chromosome. So this became our working model for how these chromosomes might form in the human genome. We got pretty excited about these SST1 repeats, and I'm going to show you a little bit more about those repeats in particular. So it turned out the version of the repeat that was on the acrocentric was a little bit different. It was about 1 kb shorter than what was found on other chromosomes in the human genome. This repeat is throughout the genome. It's not on all the chromosomes, but it's found on a lot of them. And then what Leo did was he extracted all the monomers from our one assembled human genome, the CHM13 genome, and he made a phylogenetic tree. And the the monomers from like chromosome 4 and chromosome 19, they clustered together. This indicates that they are very similar to each other. But what he found for the monomers from the 3 acrocentric chromosomes was that they essentially formed one large cluster, which really indicated that there was ongoing exchange, and you couldn't differentiate them by chromosome, ongoing exchange between these chromosomes. And so our idea at that point was that SST1 repeats, in fact, might be the hotspot, might be the breakpoint, the fragile site for these recombination events. And I told you SST1 repeats are actually, they're spread out here on lots of different chromosomes in the human genome. And kind of one of the things that Leo and Andrea noticed was that they were often also associated with segmental duplications. So we think that Robertsonians may be one manifestation of the recombinogenic properties of these repeats in the genome. And that's kind of an ongoing area of investigation, something we're excited about now. So they were often found near these segmental duplications, which are regions of near identity that form between, that are regions of identity between 2 different regions of the genome. So we'd like to figure that out. We're still working on it. But in the meantime, we put forward this working model where these PHR domains come together because of the rDNA arrays. So the rDNA arrays from 2 different chromosomes can come together into the same nucleolus. That brings the PHRs into close proximity so that they can undergo these frequent what we think are probably frequent recombination events. Most of the time in meiosis, recombination is gene conversion. So that's probably what a lot of the exchange between the non-homologous chromosomes is doing. But occasionally you get one of these crossovers, and this would be that Robertsonian event. So in the last couple minutes, I'm just going to tell you how we've been testing this model, a couple things that we've been doing in the last year. So we found in public repositories 3 different cell lines that each had its own unique Robertsonian translocation. We've got 2 that have 13-14 translocations and 1 that has a 14-21 translocation. And we validated that these were translocations using these cytogenetic approaches. And so then the next thing we wanted to do was to test the model. And the model is that we should have SST1 repeats at the breakpoint. We should have ribosomal DNA should be completely lost. It should be on this little piece that forms but won't be retained because it has no centromeres. And the Robertsonian should have 2 centromere arrays. That's the way we've drawn it. That should be what occurs. And so what we did was we collected a whole bunch of different kinds of sequencing DNA and other kinds of DNA or other kinds of data too. And Serge, and actually I think Andrea also ran this, assembled these chromosomes for the first time. And long story short, we can say that all 3 of these predictions are satisfied in all 3 cases. So it looks like SST1 really is the breakpoint. This is really exciting. As far as I know, these are the first Robertsonian chromosomes that have ever been assembled, but it lets us think about how we might expand this to other types of these events. And I just wanted to show you this really nice Centini plot that Andrea made because I think it's quite lovely. So what he did was he aligned, we've got chromosome 14 and 13 here. These are the short, remember I told you these are acrocentric, so the centromeres are near the ends. And this is kind of that little piece that I was showing you. And he aligned these to one of our Robertsonian fusion chromosomes, the assembly. And you can see, basically, you can still see the centromeres in the assembly. And then right at the junction are the SST1 repeats, which we believe are the break point. And let's see, we've also got, he did this for all of them. I'm just showing you, this is one of the 13-14 fusions. This is the 21-14 fusion. And again, the SST1 repeats, 2 centromeres, and the rDNAs, which is shown up here in gray, are lost. So I just think this is a really beautiful visualization of this result. Oh, and sorry, I covered you up down here. The last thing I'm going to tell you about these chromosomes is about the fact that they're dicentric. So we love centromeres in my lab. We think they're really interesting. How is it that these chromosomes are able to persist with two centromere arrays? If you read a textbook, it will say that dicentric should not be stable. And that's because they can attach to the pole in a manner that really rips the chromosome apart. But what is it about these chromosomes can actually be stably transmitted and inherited. And so we thought there might be some epigenetic changes at the centromeres that would allow that to happen. And that's exactly what we found. So here we're using our SPA-SIM approach again. And what Tamara has done is she's got a FISH probe to centromere 13, a FISH probe to centromere 14, and then she's got an antibody to a kinetochore protein. This is the SEMP-C protein, and that indicates where the kinetochore formation is happening. And you can see for the two 13-14 fusions, which are shown here, SEMP-C is really clearly co-localizing with SEN14, and SEN14 wins in this case. And this was actually using much different methods, but this was actually concluded long ago by Beth Sullivan. So this is very consistent with the literature. For the 1421 fusion is not quite as clear what's going on there. It's quite interesting. We've got our two centromere probes. The SEMP-C seems to be a little bit between the two of them or binding in some way to both of them. It's actually a little bit heterogeneous, even in the 10 spreads that we looked at. And all of the data we have so far is consistent with a little bit of each of the arrays being used together to form a centromere. So that's a different solution to the problem, but also allows that chromosome to be transmitted. Okay. So why do these form so frequently? We've got this working model for humans, and then if we think about out in nature, we think sequence homology is going to be really important. It could be any kind of repetitive DNA that's shared between two chromosomes. We think that bringing this repetitive DNA together is going to be an important feature of how they form. So this could be the nucleolus, but it could be other things like telomere clustering or centromere clustering that happens in meiosis. And then some kind of recombination will be important for those translocations. So then just to sum up, there are these parts of genomes that have been dark matter. We haven't had access to them, but using these new assemblies, we can start to probe their structure and their function in new ways. And that includes the centromeres and the P arms of these acrocentric chromosomes. And I'll just conclude by acknowledging my lab. They're a great bunch of people to work with, my collaborators and my funding sources. And I will stop there and take your questions.
So
I inevitably have a question or three, but somebody in the audience should ask a question.
Yeah, we have a question in the audience, Rob, and then I'm gonna show your chat.
Oh,
Go
great.
ahead.
Oh,
Go
yeah.
ahead.
Rob, you want to go ahead?
His may take a while. Why don't you go first?
Okay, so there's the PHRs and the acrocentric chromosomes with the rDNA repeats. Just by the nature of being acrocentric, those rDNA repeats are telomere proximal.
That's right.
So I think there's a lot, from a naive perspective, there's seemingly a lot of similarities to telomere maintenance mechanisms. And in particular, do these PHRs look like subtelomeric repeat sequences that may facilitate telomerase independent telomere maintenance, where you can get strand invasion from one chromosome into the next chromosome? And if so, have you guys pursued that as a possibility? I mean, PHR similarities, subtelomeric repeats, and the PRDM9 trimethylase, correct?
Mm-hmm.
Are you getting non-coding All2 transcription in an analogous fashion like you would get with tera-RNAs that are synthesized into the telomeric repeat sequences? Just spatially, these are occurring on acrocentric chromosomes, so they are physically proximal to those telomeric regions. it's really interesting. I don't know
Yeah.
how much similarity there is, or have you even considered that?
Yes, and okay, so the question is kind of, have we thought about parallels with telomeres and telomeric DNA and repeats and telomere maintenance? And I think I do think about these things because I think about different kinds of repetitive DNAs. But I think Eric and Andrea could answer better, but I'm pretty sure there's no telomere sequence in the PHRs. It's a lot of repetitive DNA, but not telomeric repetitive DNA.
Do you think it could function as substitute for those telomeric repeat sequences? Because
Ooh.
it is a shared sequence motif between the chromosomes.
I really have no idea.
Across the RDNA, this region that's called the distal junction of RDNA, and there's some satellite sequences beyond that further. And the other thing is that they are repetitive, and they do have repetitive sequence classes, but they also have genes, and they have a lot of, well, I think they're poorly annotated.
They're poorly annotated.
But
That's
we do know that there's a lot of long not coded parts. So that's what is already in those regions. Are they, do you know if they're being driven by PRDM9 trimethylase activity? Because if it's a trimethylase, clearly there's an epigenetic mechanism for nucleating transcription competent polymerase. Yeah, but those PRDM9s would be active mostly. They should only be active during
in meiosis, I think. And we are actually we're trying to look now at the transcription of these SST1 repeats. And I think it's going to be restricted in embryonic development. But that's something and that's not easy to look at because getting, embryonic information is difficult. But I think that, when they are recombinogenic is something we're very interested in, and how that depends on the chromatin landscape and how that's controlled. Yeah.
Kind of a follow-up. Cells that actually have these translocations, they actually have increased DNA content.
They actually have lower.
Do they have?
Yeah, because they lose those little pieces at the ends. Yeah, they have lower DNA content, a little bit lower.
Okay. I don't know if it's functionally relevant or not, but people looked at this. So there's a correlation between DNA content and cell size.
Uh-huh.
And I'm just wondering if there's change as a consequence of even microploidy loss, in this case, like that's loss, right?
Mm-hmm.
Because there is a correlation between the mechanisms that drive cell cycle control and increased sensitivity to cancer risk.
Right. I don't know that anyone's looked at that, but it is a relatively small fraction of the whole genome that's lost. Yeah. Yeah. I think it may actually have more implications for meiosis, because chromosome size really drives the number of crossovers. And now the Robertsonians are going to be much better at getting crossovers than the original acrocentric chromosomes. And I think that could potentially be a mechanism by which they exhibit this meiotic drive. That's pure speculation.
Is PRDM9 only expressed in meiotically competent cells?
Correct. Okay, so there's a question online.
Well, just following up on Jennifer, can you hear me?
Yes. Yeah.
So just to follow up, your comment that Iranians will increase recombination numbers per genome. I don't get that, because there's an obligatory recombination per arm on a classic human chromosome.
Mm-hmm.
In a mouse, there's only one obligatory recombination event per telocentric chromosome. So the total burden or opportunity to recombine doesn't seem like it would change much going from telocentric to conventional centromeric.
I think it's more, so like chromosome 22 and 21 in humans, they're the smallest chromosomes. And there's a substantial fraction of them that get no crossover. So it's something like 7 to 9% actually don't ever get a crossover. And they're very prone to missegregate in meiosis in humans. And so I think but now you're taking a 21 and you're fusing it to 14. Now you're making your chromosome like at least twice as big. It's even bigger because 14 is bigger than 21. And so then you're like, that chromosome is going to get a crossover almost surely. And that means it's going to segregate correctly. That's kind of that's the way I'm thinking about it.
Well, there's a distinction between the crossover of actually happening or resolving without a crossover, right? So there's an obligatory event on each arm. And that's true whether if you're a tiny chromosome, you're going to have an obligatory attempt at a crossover event. Now, whether it resolves and whether it segregates into the final oocyte or sperm is another matter. At least that's my understanding. I may be wrong, but let me get on to my question is really kind of, can you give us some technical help? So we've sequenced about 3,200 chromosomes from mice. We have really good sequence, and we're heading towards telomere to telomere for, we hope, all 3,200 of those chromosomes from 106, about 152 different strains of mice. And do you think it would be plausible to detect the Robertsonian translocations on the basis of pure high quality sequence? Or would you have to do a squash?
No, I don't think you would necessarily have to do a squash, but you'd have to know the structure, a common structure of the Robertsonian. So actually, Adam Philippi is working on a human genotyper from short read data and I think it's working. And so I think if you knew the structure, the common structure of one of these mouse Robertsonians, you could probably design a short read based genotyper to find them. But without knowing the structure, I don't think you could do it. You'd have to at least assemble one genome's worth of Robertsonians.
I got it.
Yeah.
Okay, we have our marching orders.
Yes, I think that's the next step. And I would love to see what the answer is. And
then I know Beni Masui, and I would love to talk to you about doing EQTL analysis of the whole assembly. While you were talking, I looked at SenPA to see whether it had any interesting trans EQTLs, and it has an absolutely massive trans EQTL that's right where a gene called MNDAL. MNDA is a well-known myeloleukemia, or somebody there in the audience will know a lot more than I do. But there's a massive trans EQTL right at that candidate gene. It'd be fun to get your opinion about the validity of that candidate. Anyway, Beni or I can.
Okay, you sent me a link, and so now I have my marching orders, it looks like.
Okay. All right, thank you.
Thank you.
I have some confusion. I want some clarity. For example, when 14 and 13 chromosome, they become Robertsonian. So now they have two centromeres. So during crossover, the portion between two centromeres, will that be, what will happen to them? Or they remain preserved throughout any generations?
Oh, the fact that there's two centromeres be preserved?
The portion between the two centromeres.
The portion between the two?
Centromere. What happens during the crossover, or they remain preserved across all generations?
Well, I don't necessarily know where the crossover is going to form. Is that?
Because if there is two centromeres, now the portion between them are completely locked.
Yeah.
So if crossover happens, then that portion will be broken, or it will remain as it is across many generations. The portion between two centromeres.
I have never thought about that. You mean if it's crossing over with the the normal chromosome?
Yes, through the.
Could it recover?
Because if crossover happens with another parent chromosome, so there will be some breakage in the Robertsonian, or the portion between two centromeres remains exactly same across many generations.
I think it would remain the same, but we'd love to look in a trio, in a family, and look at the inheritance, and that hasn't been done. But even to look at you'd have to catch a crossover, and those are pretty rare. So we'd have to get incredibly lucky to find that event.
And one last question. What is the role of CNPA? Is there any role found? They affect something? Or they affect the body in certain way?
CNP?
Yes.
Oh, Sempe?
Sempe. Yes.
I'm sorry, what, I didn't
Miss,
catch the
do they have
question.
some functionality?
Does it have functionality? It's a
Some
histone variant. And it specifies the nucleosomes where the kinetochore will form. So it's a histone H3 variant.
Thank
Is
you so
that
much.
your, yeah, okay.
No, just bring it back.
Am I talking loudly? Yeah, okay. So this is a little bit off topic from the Robertsonian. I was mostly interested in the chromosomal segregation and I guess aneuploidy. Are you able to track something like that in vitro over the course of mitosis, sort of like in vitro model of aging?
Oh. Like, does aneuploidy increase as the cell line passage
Yeah, I would
increases?
expect that it would, but I'm wondering if we were to give you cells that we
Mhm.
derive from two different genotypes, and our hypothesis is that one genotype will age better than the other.
Uh-huh.
Will you be able to track something like that in vitro? Would it be possible?
Well, I think so. You could either do what Enzo was saying earlier, you could do single cell DNA-seq, or you could do a whole bunch of chromosome spreads, which would not be super easy, but could be done. But probably DNA-seq would be the easiest. Yeah, that would make it feasible. You could look at thousands and just use coverage.
Except it costs a lot. But
It would cost a lot. Yes,
Okay,
there's that.
Way would be better, but I had a second question too. When I looked at the nice video you had with all the chromosomes kind of spreading apart, that looked energetically very expensive. Is there any relation with the mitochondrial genome?
Oh, wow. I have never thought about that. I don't know the answer. That is true. Yeah, I guess so. Yeah, I never thought of it like that. Hi, in the back.
Can you hear me?
Yes.
So I have a question about the Robertsonian chromosomes when they fuse to each other. You said the small portion is lost,
Mhm.
and that small portion contains the ribosomal DNA, right?
Mhm.
So does that affect anything downstream in the protein translation?
Yeah, that's a great question. Something we've thought about. There are ribosomal DNA arrays on all 5 acrocentric chromosomes. So you're really only losing 2 of 10 arrays when that happens. So a lot of people tend to think that there's more ribosomal DNA repeats than one really needs. So that's why it might be okay to lose a couple of arrays, and you might be able to upregulate the remaining copies of the genes in order to compensate, in order to make enough ribosomal RNA, but that's a hypothesis. I don't think we really know that that's how it works.
You do
Yeah.
some big variation in copy number for visual.
Yes, for sure. Yeah.
What might be a reason to cause Down syndrome? Most of RNA, RDNA, that one. But
Yeah, could that contribute to
Down syndrome, yes.
yeah, we've wondered that ourselves. Yeah.
In those scenarios, are we lost blocks? Are you seeing a compensatory upregulation for mixed signaling to RDA to increase?
Yeah, we have not looked at that. Yeah. It would be useful to be able to create these things de novo, because we imagine when we get a cell line that it's had a lot of time to adapt to its new genotype. But if we could somehow, in a very abrupt way, switch from one state to a Robertsonian state and then look, in the immediate aftermath, what happens, that would be very useful. We don't have that ability. Yeah.
No, but I'm wondering if you go back to the roots, it's much easier to play with the RDNA.
That is true. That is true.
But that underlying nutrient signaling RDNA transcription, even though the all one transcriptional apparatus is only partially conserved, there's a lot of divergence between species.
signals component
Are very
are
conserved.
very
Mhm.
conserved. So you could go back to the when you decouple all one transcription from small.
Mhm. We have those lines in the lab.
So you might be able to evolutionarily regress to evolve here.
Uh-huh.
Much easier experiment.
Yeah, we did a big screen. This is really tangential now, so people may want to go, but but we did a big screen in budding yeast. We took the whole yeast deletion collection and we just genotyped them from for rDNA copy number, like 5,000 strains or something. We did this with digital PCR, and what we found, the most striking result was that strains that had deletions that affected DNA replication had very short rDNA arrays. And so we took that to mean that if your DNA replication is compromised in some way, then you can compensate for that by shrinking your rDNA array. Because actually replicating the rDNA is it's difficult, especially the way the origins are set up in budding yeast.
Well, I mean, that would be consistent.
Yep.
Yeah, so is there a role for Robertsonian translocations and primate evolution? Have you guys looked to see what the history is?
Well,
Yeah, so this is something that I didn't put in my presentation, but I probably have a slide on it. I'm not going to try to find it. But we were very curious about the structure and organization of the acrocentric chromosomes in the primates. And now because there are these T to T assemblies, we went in to see, I think the PHRs are still being defined. I don't think they're quite defined yet, but we looked just at SST1 repeats and their organization, whether they're inverted and on which chromosomes and which chromosomes have rDNA and SST1. And I guess to summarize it, what I can say is don't lay awake at night worrying that non-human primates get Robertsonians. The human genome is essentially, it's like a perfect storm scenario with that inversion and where the rDNA is located. That setup to form that type of Robertsonian, the SST1 based Robertsonian, does not exist in chimps or bonobo or gorilla or orangutan or gibbon. So it's very unique to the human genome. Yeah, a lot of people ask that question, and it even came up on the call just yesterday. It is a Robertsonian, but it does not appear to be an SST1 mediated Robertsonian. Yeah, it's a chromosome fusion, but probably not the same mechanism. So there's lots of other mechanisms. I'm just telling you about these common, they're called common. There's probably lots of other kind of one-off mechanisms that don't necessarily share the same formation. Yeah.
Oh, that was the question. It's like, that'd be interesting to see how, from an evolutionary perspective, how many of these events occur in humans. Like, I think we're just at the beginning of understanding them. So that was the question.
Yeah, it is. I mean, these fusion chromosomes are, like I said, even, 15, 20 years ago, people recognized that this was the most common type of karyotype evolution in mammals.
That's what we learned.
So SST1 is like at least a little bit of the story, but there must be other players and other events that we don't currently appreciate.
And if people's not too tired, it's just like a little curiosity about I might miss some biology of the segregation here, definitely. But question is like for chromosomes with two centromeres, in some cases, they might resolve in funny way, like, because like some centromeres, like the ideal case is when centromeres and on the same chromatid just get on the same side of the mitotic spindle, right? And this can happen in a certain number of cases. So I think, I don't know why you worry a lot. I mean, it seems to me we don't have to worry too much if this happened or not, because like sometimes can happen, sometimes cannot. So just like it get just discarded. Whenever it doesn't happen, the cell just don't proceed. So I don't see how am I missing something here, or is that the case? Like, do you think sometimes they segregate correct? Sorry, what I want to say is that to me, there's a certain chance for them to segregate correctly, even if, they have two centromeres, and that's enough to guarantee like species survival, like in other terms.
Yeah. Well, I think like if they were both active and kind of competing for microtubule
That's the other
capture
thing, yeah.
in mitosis, I think eventually you would have, even if your error rate was like 10%, that would be too much. Yeah, I don't think you could make an embryo or a human under those conditions.
So we just select.
So yeah, but I think it's certainly true that in the meiotic divisions in the oocyte, I think that's where they see this meiotic drive, and that's because you have polar bodies. That's why you only get it in females, right? So like a centromere can win and a centromere can lose.
Yeah.
But that's very specific to the polar bodies, where the loser goes into a polar body and you still end up with a germ cell in the end. Yeah. Okay. Thanks everyone for hanging in there.